Hello.
Hi.
Hi, how are you?
Yeah,
How's
I'm
everyone today?
I'm eating
just
peanut butter toast.
Oh, okay.
Mm.
So, welcome to the meeting. Is there anything anyone wanted to talk about or present before we begin, or talk?
Um, I went to that APS physics symposium, and there was one on networks that I'm going to send you. She did work with zebrafish, and she pulled on the tissue of the developing dome, the first one of the first stages in the zebrafish, with a pipette. And she found that the um, tissue that was on the sides was more firm than the top. And then they looked at it through a microscope and looked at the tissues and then made a network out of how the tissues were joined. If the cells were joined together, then they joined them in the network, and if they weren't, then they left them off, and they made these nets, and then they analyzed the nets. So,
All right. Yeah, that sounds great.
yeah, that's one of the things. There are a couple more, but I'll save them for another day. But that one was embryology and networks.
All right. Yeah, that sounds great.
And I'll get the references for next week. Just
Okay.
I was downloading COMSOL while watching APS physics all last week, and I almost fried my brain.
Oh, yeah.
Anyways, yeah.
Well, thank you for that. That's great. I'll have to look into that a little bit more. Does anyone else have anything, or?
Hi everyone, I'm Abhipsha. I'm joining these meetings for the first time, so just want to introduce myself.
I'm having trouble hearing you.
Oh, sorry. I just wanted to say that I'm joining this meeting for the first time, and so I just look forward to it. Hope that was audible.
Oh, welcome. Yeah. Yeah. Are you interested in GSoC?
Yeah, I came from
Okay.
GSoC.
Well, that's good. Yeah, thank you for attending. Welcome. Yeah, we'll talk about that in a little bit, and then we'll probably talk about some other things. Our meetings are usually pretty broad, but if you're in our Slack, in the OpenWorm Slack, and the DevoWorm channel, you'll get news about the project. As we go through the application process, I'll be posting more there. Okay, thank you. Karan or Harikrishna, did you have anything?
no, nothing in particular.
Okay.
I'm still working on. I'm still making progress.
Just wanted to confirm about the deadlines.
Yeah.
I think it's starting from April 4th, the proposal period. So just finishing some changes. I'll be sending it across this week. I think at least the first draft. Just have some last minute changes that I'll edit and send
Right.
across.
Sounds
Yeah,
good.
that's a recent update. Yeah.
Yeah. Hello, Ujjwal.
Hello, hi everyone.
Hi. How are
Hi.
you? How are you doing these days?
Like last few months are very busy, so I was a little bit distant. But I guess now I can start regularly attending meetings once again.
Okay.
So I was working on my bachelor thesis project, and there is a side project that we are doing with our professor to change to create some sort of new navigation, which is still under patent, so I cannot talk much about it.
Yeah.
So that's it from my side.
Well, that's good. Welcome back. Yeah. Glad to hear you're doing
Thank
well.
you.
Okay. And EBD, how are you?
Hello.
Hello.
I'm fine. How about you?
Pretty good.
Nice, great. Sorry I missed the previous part. I just joined.
Oh, just people introducing themselves and talking about some things. You joined the. I see you joined the Slack for GSoC.
Yes, sure.
Okay. Yeah. So that's good.
So if you want, I can introduce myself again.
Yeah. Go ahead.
So, yeah, my name is Muhammad Abdullah, and I'm currently a third year. So when I joined the OpenWorm, I was second year. So now I'm a third year from VNIT Nagpur, and I'm currently learning object detection and semantic segmentation along with just started the GNN part. So that's why I just got the attention of the project.
Yeah. Welcome.
Thanks.
So, all right. So, why don't we start with GSoC? I know a lot of people are here for that. And again, I like to go through this weekly so that people have an idea of what we're doing. So this is the website here. This is the timeline for 2022. So, let's see. The contributor proposals are open on April 4th. So we're somewhere in here. And the next deadline, of course, is when the proposal portal opens. So when the proposal portal opens, that's not so important. It just means that they start accepting applications. The deadline, however, is, of course, April 19th. So that's in about a month. So you have 4 weeks or so to write your proposals up. And I talked about the proposals last week. I talked about the outline of proposal, what a proposal should look like. It should have the introduction, motivating what you're doing. Then it should have the maybe some sort of proof of concept or some sort of details about the method. If you can work with some of the data, that we have, it's better because you can show how your method is going to work. Sometimes with some of these methods, you have to tune them, play around with them in advance to get a sense of what they can do. So you don't want to propose something that's wildly off base. You want to be able to have a good proposal, because when you start the program, then you want to jump right into it and you want to be able to hit your goals. And so that last part of the proposal is a timeline that has, everything week by week. What am I going to do every week? And, you're going to have to sort of know what you can do in that amount of time, what targets you can hit. But also, if something goes wrong, you want to be able to adjust for that and do something else while you're waiting on that stumbling block. So I want to make sure that people have that in their heads, kind of the deadline, and then how to outline everything. And, and you can send me a proposal draft if you're, when you're at a good stage. Don't send me something, that's very loose set of ideas. Just send me something that's a draft that I can look at and review and give you some feedback on. It's going to take a little bit of practice, especially with the timeline, to get it right, because you'll have to. Okay, so we have someone else is joining us. So she's going to be back in a minute. So that's I wanted to make sure that everyone knew that if you're applying this year, the deadline, and then how to write a proposal, and then that I'll look over and review them as you go along. So these are the projects here. This is 22.1. So this is the NeuroStars portal. It's run by INCF. That's our sponsoring organization. And so if you go to neurostars.org, if you haven't gone there, you can log in and you can ask questions in this forum. This is kind of, runs parallel to our Slack. Our Slack is private to our OpenWorm group, but INCF is broader, and a lot of times people will come here to find out about the project to see what projects are being offered by INCF. So this will have like sometimes people will post questions in here, and then I can answer them. But you can also do that in the Slack. So if you see this part, what I can do before GSoC, what can I do before GSoC? Hello. Okay. What can I do before GSoC? You can ask one of the mentors to direct you to the data source, and you can start working on it. So this is just basically going to the different data sources that we have. And if you have any questions about any sort of data that we might have, I showed DevoZoo in a previous meeting. So that's going to be valuable. I'll post the DevoZoo link in the chat or in the Slack. And that's one source, but there are other sources of data. Just basically ask me for the about your question and what data source you would like to have, and I can point you to it, because DevoZoo has a lot of different sources of data, and I don't know, you might just not get the right one. There's certain types of data that are better than others to work with when you're starting out. So we have a website here, preprint on DevoLearn, which is a platform we developed in the last couple years of doing GSoC. It has a lot of components to it for doing like pre-trained models on embryos. And then DevoWorm AI is actually something Ujjwal worked on for his project, and that shows a lot of our different software packages that we have. So we've done a lot of work on working on machine learning and deep learning to analyze microscopy images. So those pieces of software here at DevoLearn and DevoWorm AI, those are two that we've been working on over the last several years and bringing those resources together. So if you're working on the GNNs, that would be added to this library. So you would be contributing to that area of research. And so then we have this other project. I don't know how many people are interested in this one. I know at least one or two of you are. And this is the digital microsphere. So this is based on Susan Crawford-Young, who's here today. She built this special type of microscope that takes images from all angles of the embryo, So you have this spherical space of data. So you have all these different angles of the surface. And the idea is to build like a framework to bring stitch those together onto a sphere. And so if you're interested in that project, we're working with axolotl embryos, and Susan has data that we're working on getting people so that they can do this project. And there are a couple of readings here, and the skill there is just kind of like, can you build a 3-dimensional or a 4-dimensional model actually, but can you project data onto a sphere? And do you have a method for doing that? So those are our two GSoC projects. So Susan right now is holding up the ball microscope, and this is sort of what it looks like. So the specimen goes inside of it, and you can take pictures from how many different angles? Like 9, I think.
I've got it so that it's there's 10 right now.
Okay.
Because I put a top one in as well as the bottom, and then the 8 sides. The 8 sides are all at 90 degrees to each other, and the pencil microscopes just fit in here and are held in place. And then you can adjust the angle here so that you can zoom in on the spot in the middle, which is, I don't know, whatever you're taking a 3D image of. And it could be anything from a snail to well, in my case, I want to take pictures of axolotl embryos, which are basically a round object in the center of this. And they're digital microscopes. So you're getting images from all sides taken sequentially. at the rate, well, the way I've got it set up at the moment. Anyway,
That's great.
So-
Yeah.
that's it. Anyways,
Thank you for showing that. Yeah. So yeah, that's the apparatus for that one, and like it's either project is good, and I know a lot of you are interested in GNNs. For the GNNs, again, we have the DevoWorm or the DevoZoo data source. So that data source is I'll put that in the Slack, and it's just like a lot of microscopy images from different sources. And I know from years past that you have to try maybe a couple different data sets to get what you want. So some data sets are really good for just kind of taking them and segmenting them with a really simple algorithm and then going from there. So you can actually, one of the things you need to do to be able to segment things, and in some of the other projects this was a big part of it, but I don't know about the GNNs. It might not be so much of a big part of it, but it's actually finding the boundaries of the cells. So in an embryo you have cells that have boundaries, and you have to define, when you get the digital image, you have to define where the boundaries are. And it's not so easy because, there's a lot of, you're really looking for breaks in the coloration. And so sometimes that coloration break is not obvious to the algorithm. If you use something like a fluorescent microscopy image, which we have and we've used in the past, which have worked typically a lot better than some of the other types of imaging, even that can be a problem, and you have to come up with ways around that. So, those are just some of the difficulties of working with microscopy data, but I think it's something that we've overcome in the past. We have techniques for doing that, so it's not a problem really. We can figure it out. But really, the data, you want to just pick something that you can do a proof of concept on, even for the project, really, because you could throw a lot of data at it, but you just want to show how it works. I want to be able to run the software. I want to be able to take some data, put it in, and get something out. So that's what you want to do. And we have, like I said, we have a lot of tricks and tips for people if they're stuck on segmentation steps, because that can be something that can hold you back. But when you do the schedule for your project, you'll want to start with, ideally you'll want to have worked with the data a little bit, know kind of what data sets you're going to use, and then you're going to go to this. I'm going to implement the algorithm in this platform. It could be PyTorch. I'm going to then maybe experiment with it a little bit, get a demo running by week 4 or 5, and I'm just giving you some ideas that might be a little late to start doing demos, but and then kind of run through and then have some goals at the end, the software you're gonna you have to, at the end you have to have a packager that the people at GSoC can run. That's basically the goal here. You want to have a complete software package that you can host it on a Gist, or you can have it as something on your GitHub, but they have to be able to download the software and run it successfully in order for you to pass. So you can't just say, this is what I'm going to do in the future and I'm not done yet. You really have to get it done in that amount of time. So that means that you have to be you can't try to solve the world's problems in this project. You're just going to have to come up with the shortest route to getting to a piece of software and then releasing it. And so that's the challenge, because and I don't know I can't remember how many weeks you have total, because there's a community period in there, but it's 13 weeks or something. That's not very long to do that. So any questions at this point?
I have a question.
Okay.
Are the timelines flexible, or is the project 175 hours, or can it be extended? Because I know this time around some GSoC projects are have longer timelines. So just
Yeah.
wanted to know that.
I think both of these projects are 175 hours. So I think that should be enough. The ones in the past we've done on the like on the pre-trained models, that was 175. Because it's really just this one part. If 350 hours would be like if it had two parts to it. But I think, that's something you have to consider though when you decide how you're gonna implement this. So, do we want to like have this huge program with a lot of bells and whistles, or just a little small package that we can use as a starting point? Take a look at the DevoWorm AI and the DevoLearn, because that should show you what kind of program you can do in 175 hours. So that's, just planning out what you need to do, planning for that size of an application. That'll give you some pointers. And again, it's on the Neurostars site. All the links and the information about the 175 hours. Okay. Thank you. Yeah. Any other questions? Right. Yeah, again, if you have questions, you can drop them to me in Slack, and you have some comments in the chat. Okay. It's okay, yeah. If you have an audio issue. You can type in the chat if you have an audio issue too. So feel free to ask questions in the chat. And then Ujjwal says, feel free to contact me over Slack to get help, advice on proposals. If you want to know more about GSoC projects in general. Yeah, Ujjwal was a student in 2020. So he was involved in the group the year before. And I think maybe a year and a half before he was successfully chosen as a GSoC student. And then he worked on another project that resulted in a book chapter. And then he worked on his GSoC project, which is the DevoWorm AI. And then, now he's doing some really good things. But he knows about the process. So if you want to ask him, he's in the Slack as Ujjwal Singh. So you can, search for his name and add him if you have a question. Or at me. So yeah. Okay. Well, if there are more questions, and you can leave them in the chat here if you want. I'll come back to them in a bit. Why don't we go on to some other things? And so a couple of things I found that are interesting this week. I know Susan had mentioned that she went to APS and she did the biophysics, a couple of biophysics sessions there. This is something I found from APS, but it is like a prize that they're running. So they have this gallery of soft matter. So this is where they take pictures of soft matter or structures, or I don't know exactly what it is, but basically there are different projects that involve soft matter. So soft matter is where you have, something like tissues or soft robots that are, different materials that are soft. So, we usually think of mechanical systems as being hard and biological systems as being soft. In this case, it could be a mechanical system that's soft, or it could be a living system that, or a mechanical system that acts like a living system. They're just, there are different ways that people define this, but this is a nice set of projects that they have selected as the winners for their thing here, their competition. So this is the first one, inflatable muscles for soft robots. And so one day while fabricating long, thin rods of a silicone elastomer, graduate student Trevor Jones said that he and his colleagues at Princeton had a happy accident. While the elastomer was still in liquid, air found its way into one of the molds. The air formed a bubble that rose to the top of the rod. When they removed the rod from the mold, they found that the bubble had hollowed part of it out, leaving the rod along off-center cavity. So this is, I don't know if you can see this, fluid-mediated soft actuators. That's their project. So this is really loud. Okay, I think that's a good gives you a good idea of what it is. So that was something that it was a mechanical system that was made of a soft material, and they used compressed air to sort of manipulate the cavity and then get this sort of actuation that kind of looks like it's moving around. Looks like some fingers grasping something, or like a worm or something like that. So it's very interesting what you can do with soft materials. It's not a biological system. This is I wouldn't call it a mechanical system, but it's a synthetic system that can be done. You can do things. You can program it to perform all these different movements. So you're just using air to inject it with different amounts of air and at different pressures, and you can program it in different ways to do these types of things. So they're able to do a bunch of experiments here, and that's that project. Then there's the creeping fingers, which is something that was let's see. So this is behind the interface. Let's take a look at this one. I don't want to have it too loud because I might have music on it again. So the way that the concentric rings of fluid flow outward in a system undergoing a Saffman-Taylor instability reveals dynamics over the unexpected length scale. Let's go a little bit further out here. This is just kind of the intro. Okay, here's the experiment. So we have this fluid flow. And they have a nice little horror theme here going, so. And they're setting up the experimental apparatus here. And so now you're able to create these patterns. And these patterns kind of look like, maybe like a bacterial system or some sort of chemical reaction. It's a chemical reaction, of course. But it does kind of look like some sort of pattern formation, as you can see. And you can just see how this pattern formation proceeds. So this is from a simple chemical reaction. And there's a lot of complexity there that gets revealed. Then there's ceramics, liquid crystals, and bubbles. And then, so these were also some things that won prizes. These posters showed the effects of bubbles on shape of an elastic ribbon, the release of material frustration in a structure made two types of clay that shrink by different amounts when fired. And the structural there.
Got that working. And I have a bunch of, I've got the relays and I've got the Arduino. Now I need to find the time.
Oh,
I'm a PhD student.
yeah, you mentioned it before.
So it's like, but anyway, I'll keep trying.
Yeah.
I'll ask if I can have a week off and then I can work on it. But first I have to get my project together.
So it looks like I got disconnected here.
Okay,
Sorry.
Bradley.
Yeah,
You're here.
sorry. Yeah. So I did this, I presented this thing on the soft
Oh,
physics.
we can't
Did you see
hear
that?
you. Oh, you're off. Your sound is off. That's
Okay, I'm here. Yeah. So yeah, did you see the soft matter physics thing? I didn't.
What, for APS physics?
Yeah.
Yes, I did. Yeah.
Okay. So
I have biophysics.
did we have any questions about that, or just?
It was a Friday afternoon, really good session. And I'll put something together about it, but I would rather go from the people's papers rather than what they did, because I would start doing screen captures once in a while.
Yeah.
Their presentation is not proper. Ugh. So I'll find their papers, I'll put something together. I'll try for next week because I don't want it to leave my head. Like, it was very good.
Well, that's
Excellent.
Good. Yeah. Yeah.
Yeah.
Okay. Let me share my screen. Hopefully this works. Right. So there's this other thing that I found here, which was this article on the animal's behavior is mechanically programmed. So this is a swarm. This is what we call kind of like swarm-like behavior. This is the animal Trichoplax adherens, and it moves in response to its environment with agility and seeming purpose. Yet it has no neurons or muscles to coordinate its movements. New work shows that biomechanical interactions among the animal's cilia are sufficient to explain how it moves. And so this is a nice, it's collective behavior in this organism. It's basically these biomechanical interactions amongst the cilia, and it's moving the organism, and it doesn't necessarily have a brain. It's just moving like this. And so the biophysicist Manu Prakash vividly remembers the moment late one night in a colleague's laboratory a dozen years ago when he peered into a microscope and met his new obsession. So it's this amoeba here. And so this is a flattened multicellular blob, only 20 microns thick and a few millimeters across, with neither head nor tail. It moved on thousands of cilia that blanketed its underside to form the sticky, hairy plate that inspired its Latin name, which is Trichoplax adherens. And so this is a marine creature that has an entire branch of the evolutionary tree of life to itself, as well as the smallest known genome in the animal kingdom. But what intrigued Prakash most was the well-orchestrated grace, agility, and efficiency with which thousands to millions of cells in Trichoplax moved. And so this is an example of like something that you would have, like a microscopy image that people take. I mean, people take these things in time series, so you can get a sense of movement of the different cells and cilia and other types of structures in the organism. So you can see some of the challenges here maybe of segmenting something like this, where you have to find the boundaries and you have to find some of the other details of the form. And so the question is how do you characterize this movement? How do you characterize the different cilia and cells that are existing in this organism? And so this person wrote a number of papers on this, and it's really interesting because we've been working on this stuff with diatoms and looking at movement in diatoms, and we've been looking at different models, and so there's a really interesting literature on like explaining movements with physical models. And so Manu Prakash wrote a series of papers on this, on how to characterize the movement of these colonies using some really interesting physics. So I'm not going to go through those papers right now, but I just wanted to pull up this article and go through it.
He did a presentation in APS physics that I watched.
Really? Okay.
Oh, it was very good.
That's great. Yeah. So he put out a trio of preprints, over 100 pages of work, and this is on the archive. And he and Matthew Stormbull, who's his collaborator, showed that the behavior of trichoplax could be described entirely in the language of physics and dynamical systems. So this is a type of model that we would use if you take the data from this organism and you break it down into cilia or cells or movements. You can then use a model to analyze the data. So if in one of these projects you're creating the software to take these images and decompose them into numbers basically, and then we can use those numbers to plug them into other types of models like dynamical systems models or other physics models, and we can analyze the movement, we can simulate the movement and all these other great things. So when you do this kind of a project, it can lead to other things down the road. And so mechanical interactions that begin at the level of a single cilium and then multiplied over millions of cells and extended to higher levels of structure fully explain the coordinate locomotion of the entire animal. The organism doesn't choose what to do. Instead, the horde of individual cilia simply move, and the animal as a whole performs as though it's being directed by a nervous system. So this is an example here of this is.
For the last 7 years, researchers at the Prakash Lab.
This is a 6-minute video. I'm not going to go through the whole thing.
And ocean.
But this kind of shows you some of the. This shows you this organism.
Trichoplax was able to coordinate movement, strategize how to find food, and even reproduce by ripping itself in two. But how can Trichoplax exhibit these complex behaviors without a brain to call the shots? The answer could shed light on the origins of the nervous system.
So that's, I don't want to go through that too much, but that's the idea. It kind of goes through why they're doing this. This doesn't have a nervous system, so they want to see how this organism can generate these complex behaviors without a nervous system. And so here's a quote. Brains are overrated. A brain is something that only works in a very specific context of its body. In the fields known as soft robots and active matter, research has demonstrated that the right mechanical dynamics can be all that's needed to accomplish complex tasks without centralized control. In fact, single cells alone are capable of remarkable behaviors, and they can self-assemble into collective systems, such as slime molds or xenobots. Xenobots are these interesting kind of organic robots that people have made. Slime molds, of course, are these collective collection of cells that ooze around in different wet environments that can achieve even more, all without the help of neurons or muscle. So this is an interesting, this is a multicellular animal, and they want to see if this happens as well, and this is exactly what we find. So this is a picture of Manu Prakash giving, he's a biophysicist at Stanford, and he's showing the Foldscope, which is a nice $1 microscope that he's created. And he's kind of talking about this organism. So here's a side view of Trichoplex where it's crawling along a surface with its cilia touching this surface and the cells up here taking a ride. So it's walking. It looks like it's walking, but it's doing it without a brain. It's basically coordinating all these collective movements in a way that allows it to walk, almost like something with a brain might walk, but it doesn't have a brain. And so that's the idea here in this paper. So we usually think when we have something going on like that, like when we have an internal clock-like signal, it's saying, okay, go, now stop, that's controlled by a brain. That's not what's happening here. The cilia aren't getting paced. There's not some central thing that's saying, go, go, go. It's the collective mechanical interactions that are setting up something that goes, goes, goes. So we're working on a paper, Dick Gordon and I, and Jesse Parent, who's also someone who works in the group. And we've done a thing on diatoms, where we've kind of proposed some of, I guess, similar ideas. It's a little bit different. But I think next week I might show people this paper. So, we're kind of finishing it up now. I didn't want to do it right now because it's still kind of too fresh to go through. I think I've shown it before in the group, but it's much different now. So you're dealing with a lot of the same issues. So any other questions at this point? So, yeah. So let me go. We also do like different submissions to things. So our community here, we do a lot of sort of collaboration. And so we have all these tasks we have this task board that's kind of like this big snarled mess in some ways. But it just basically, when we have these meetings, we like to talk about different things. And sometimes they move off into the ether and you never hear about them again. But one of the things we like to do is use GitHub to organize our meetings. So this is our group meetings repository on GitHub. Here's our DevoWorm group. If you want to follow our DevoWorm group on GitHub, that you can find out more about some of the things we do. But our group meetings repository actually has a lot of this stuff. It has a lot of issues on our issue board that we've talked about in the past. So we talk about a wide range of things in the meetings, from talking about diatoms to talking about network theory and network science, to talking about embryos and general development. There are all sorts of things that we have on here that people are doing in different phases of doing. So if you want to check this board out, if you see something you really like, you can ask us. You can make a comment on the issue or ask about it in the Slack. Some of these things are out of date. They're constantly, we're not updating it like we should, but there are a lot of interesting ideas we like to put here just so that they don't go off into the ether. And so that's something else. I'd like to finish up today with some papers. This is the mechanical programming stuff. Oh, actually, I think we had a comment here. So we have two comments. Okay, so let's see. ABD, nice. I didn't know there were USB microscopes as portable. Yeah, that was in reference to Susan's microscope. On says, this claim in the paper largely depends on how behavior is defined now. It seems like the trichoplex is interacting with its environment, much as a plant, a multicellular organism, interacts with the world. We classify the plant's interactions with the world as behavior. I'm not sure. So was that the question you were going to ask? Okay, yeah. So yeah, that's a good question. I guess behavior is an interesting thing. I guess if it's moving, people call it behavior. Sometimes it depends on what it looks like. So if something looks like it's moving or it's like animated in some way, people will claim that's behavior. In plants, I do know there are people who study plant behavior. It's not a very common field, but people do. And, the definition, I guess, of behavior is if it's moving, if it's interacting with other things. And so, it's a very clear thing when we have an organism with a brain. We know that, like, if the brain's generating it, maybe it's behavior or something like that. But if it doesn't have a brain or if it's a plant, then it's, actually we know if it's a bacteria that it's behaving, if it's moving around and interacting. So that's also somewhat clear. But then, we get to these gray areas where is it behavior? And I guess it's behavior. I don't know. So a lot of the stuff that people do is they actually look at things and they say, does it look like something I'm familiar with? So if it could be behavior if it's doing nothing. If there's a lot of biochemical interactions or signaling interactions, for example. But we don't generally classify that as behavior. So, yeah, I think that's a great question. Thank you for asking. I know it wasn't a very clear answer, but I don't think there's a good answer for that, and it's not because it's not a good question. It's yeah. So Ujjwal says, adding on how this mechanical behavior is any different from the distributed consciousness shown in bees. Well, that's another good question, and it's really not that different. There's this area called swarm intelligence or collective behavior, where people have tried to characterize some of these behavioral patterns, where you get these interactions and you get these things that form these complex shapes and complex patterns amongst like a lot of organisms or a lot of cells or something like that. So yeah, it is very similar to what you see in ants or bees. I mean, there's a huge literature on collective behavior and on swarm intelligence and things like that. And so the mechanical behavior part, though, people have looked at swarms for centuries and said, well, that looks intelligent, that's interesting. But they've never really been able to characterize the interactions. And so a lot of these physical studies are ways to sort of characterize what's going on. Like, say, that there are rules to this, or that there's certain physical conditions that result from this. So a lot of swarm intelligence can be characterized by the interactions, but they can also be characterized by the physics of the swarm itself. And so sometimes people have done experiments, for example, where they've looked at the sort of the physical properties of the swarm. Ants will build rafts by linking themselves together and forming this huge mass of ants. And they can ride on floods into different places. And that sort of collective behavior requires certain physics inside of that group of organisms. And so there's a physical aspect to that. So it's very it's not well known. That area is still pretty open. I think people are starting to get there, but I don't think it's anything really definitive. So let me go back to the papers. So the paper, the first folder had these papers on mechanical programming. And so this paper is one of the archive papers. This is a versatile pattern this isn't the right one. This is it. So this is a swimming rheometer, self-propulsion in a freely suspended swimmer enabled by viscoelastic normal stresses. So this is going from like the popular article to some really technical words here. And they're proposing that this is a rheometer. And so you have this problem where you have self-propulsion at low Reynolds numbers. And a Reynolds number is simply the drag that the water produces on the organism. And so it has to be able to propel itself in this way. And this is a concept commonly known as the scallop theorem. Here we present a truly self-propelled swimmer, force and torque free, that while unable to swim in a Newtonian field due to the scallop theorem, propels itself in a non-Newtonian fluid as a result of fluid elasticity. So this is a non-Newtonian effect. This propulsion mechanism is demonstrated using a robotic swimmer. So they were able to build a robotic swimmer comprised of a head sphere and a tail sphere, where the swimming speed is shown at reasonable agreement with some theory and some simulations. And so then they also have this image. They do some imaging. They show the propulsion of the swimmer is driven by a viscoelastic jet at the tail, which is the tail end of the animal. And this develops because of some of the asymmetries in the body of the swimmer. And so they're able to look at the geometry of the organism, and they're able to look at how it's moving in the water and show that that organism is actually generating the conditions for its further propulsion. So that's kind of really interesting, kind of interactions between the physics and the organism. This next paper then talks about excitable mechanics embodied in a walking cilium. So this is where they look at, the abstract reads, rapid transduction of sensory stimulation to action, which is what we see in the nervous system. It's taking things from the environment, stimuli, and converting it into something in the nervous system that then converts it back to some sort of movement, some sort of action. So it's what the nervous system does. It takes in sensory input, it transforms it into some sort of thing that can be affected into the environment in terms of action. To this end, most animals use the sub-second excitable and multi-stable dynamics of a neuromuscular system. So the neuromuscular system like ours, where we have inputs and outputs in terms of movement. Here, studying an animal without neurons and muscles, we report analogous excitable and multi-stable dynamics, embodying the physics of a walking cilium. So they define a walking cilium as a phenomenon where the locomotive force is generated through contact with a substrate which is periodically reset by steps in the direction of motion. So this is kind of like, I don't know if people are familiar with a random walk or a random walker, but this is similar, where you have this thing that kind of moves in random directions, and then those random directions, I guess they're reset periodically so that they kind of move not just kind of randomly in kind of a cloud, but they move maybe in a certain direction. So we begin by showing that cilia can walk without specialized gait control and identify the characteristic scales of spatiotemporal height fluctuations. With the addition of surface interaction, we construct a low-order dynamical model of this single cilium subunit. Then they talk about the collections of these walking cilia. So like we saw in the video where you had all these cilia underneath the body. En route to an emergent model for ciliary walking, we demonstrate the limits of substrate-mediated synchronization between cilia. So this can, they show evidence of a localized multi-stability of the different cilia. So there's this, they can exist in different states, and there's local synchronization. And so there's this out of equilibrium mechanics, and this directly controls the locomotive forcing of walking cilia. So this is where you get these, we can show a minimal mechanism, what they call trigger waves, by which these walking cells may work together to achieve collaboration, such as coordination of hunting strikes across 10 to the 5th cells, which is it 10,000 or so, without central control. So this is really a nice paper. It shows like these, actually it shows an example of the Fitzhugh-Nagumo oscillator, which is an oscillating system that is, it's a model for like neurons and oscillating neurons and how they synchronize. And so they show this in a system that doesn't have neurons. So then there's also this paper, Mobile Deficits Born from an Energy Cascade Shape, the Locomotive Behavior of a Headless Animal. So again, there's this headless aspect where it doesn't have a brain, but it does have this sort of behavior that looks like it is generated by a brain. And so, again, this paper talks about some of the physics of it. I don't think there's anything special about this paper, but it kind of talks about some of the modeling that they do. They use this geometric model. And then this one is ciliary flocking and emergent instabilities enable collective agility. So this is again where they don't have muscles, they don't have neurons, they don't have brains, but they have this sort of coordinated behavior. This is what they call ciliary flocking. So this is where they have these collective ciliary behaviors that can move the organism in different directions. So this is something they call an active elastic resonator, and they use this model to look at some of these things that this organism is doing. So it's very interesting work, and so you think about like some of these constraints that we typically think about. We say behavior is generated by a brain, but maybe bacteria have behavior and it doesn't have a brain, or plants could have behaviors. They move all the time, but they don't have brains. And so actually it's the physics of these systems that can explain a lot of this. And in our paper on diatoms, we talk about maybe some models from neuroscience that might explain this, but models from neuroscience that don't really have any neuronal content. So that means that you can use sort of the similar models, but you're replacing neurons with other types of cells or other types of biological structures, and you're using those to characterize that behavior. So that's what that is. Any questions at this point? All right. So I might do one more paper before we end, and if you have to go now, is the top of the hour. We usually go for like an hour, and then maybe we have something right after the hour. So we don't have any more questions. I'll continue with one more paper, and then maybe if you have any questions to end the meeting, maybe something about GSoC or something else, then I'll be happy to answer them. So let me go back to this. I want to talk about that today. I'll talk about this one. We've had this one sitting around for a while, and we haven't talked about it. This is something that we've been interested in the group for a while, but we haven't really found the right way to approach this problem or to write it up. This is Synteny breakup in chromosomal chaos. So Synteny is where if you go to a genome and you have a chromosome, that chromosome has an order of genes on it. So we're familiar with how genes are organized on a chromosome. So if you have like if you have a linear chromosome, and like in bacteria they have a circular chromosome, but in most vertebrates and in invertebrates they have this linear chromosome, and you have different genes on it. All right. And so the idea behind Synteny is it's the gene order. And so, you might have A, B, C, and this is very simplified. I just wanted to give you an example. This A, B, C, D order is called Synteny. And so, that's great, because if you're trying to transcribe genes, the promoter regions up here need to know what gene this is. And then if you have genes that are next to one another, they're physically linked, sometimes that's important for function or co-expression. Those things can be expressed together. And then, they tend not to be moved around during recombination or anything like that. So this gene order can be quite important sometimes for maintaining developmental programs where tissues form in a certain order and genes are expressed in a certain order. So it can be quite important. We talked about Hox gene clusters before. Hox gene clusters are similar to this, where they have an order of different Hox genes. And that order is very important for maintaining segmentation in an organism. So that's good. But sometimes that can be ripped apart. And so there is, of course, recombination where different parts of the genome flip. They flip their position. So let's suppose that in this one, in this linear chromosome, these two sections flip. And what that means is that A and B are down here and C and D are up here. So if we have this chromosome here.
Does that happen as a result of radiation?
No, I don't know why like you get the sort of Synteny translocation. Sometimes it's just that like things recombine randomly in the genome and they get flipped. A lot of times though things are conserved if they're close to one another. So if you have two genes that are next to one another, the probability of them getting recombined is low. There's no real causal mechanism other than like random recombination. I'm not sure. There may be other reasons why you would have a breakup of Synteny. It could be like transposons sticking themselves in. There are many different reasons why it might be. I don't know, but I don't think it's radiation. But anyways, so this changes the order, and so now this has consequences for how the phenotype is expressed, because maybe now say if these are Hox genes, your Hox genes are in different positions, and your segmentation can be mucked with. So there are different mutants of Drosophila where you have different sort of Hox gene mutations or Hox gene rearrangements, and those are not, those are sometimes they're not functional mutants, sometimes they're very special mutants with special phenotypes. So, this can happen in a number of ways. You can have different types of Synteny breakup, and so this ordering is important in the genome. So the idea, though, is it's very important for development, and I just wanted to go over that concept to go over some of these papers. So this is what they call chromosomal chaos, and if I can find this, okay, here it is. So this is something they call chromosomal tectonics, and so this is early animal evolution. This works. All right, I think it's going to stick on me here. I don't know why. But let me see if all right, there we go. Let's try it again. All right, there we go. So this is from Quantum Magazine. This is Secrets of Early Animal Evolution Revealed by Chromosome Tectonics. So large blocks of genes conserved through hundreds of millions of years of evolution hint to how the first animal chromosomes came to be. So blocks of linked genes can maintain their integrity and be tracked through evolution, meaning that you can see these genes in the same order in a lot of different organisms that have a common ancestor. Then discovery of its foundation is what is being called genome tectonics. So this is an idea that it's kind of like plate tectonics, where you have these continental plates that are drifting along the surface of the earth, and that they don't really, they're always there. They're kind of there over geologic time. They build up from cratons, but they're there for longer periods of time. So they're continents just don't break up at random. They're really kind of conserved, and so this is the idea they're talking about here. But the thing is that these, I guess if you want to call different gene orders tectonic plates, sometimes they can break apart, and that's why they call this genome tectonics. So chromosomes, the bundles of DNA that star in the mitotic ballet of cell division, that's a little poetic, play a leading role in complex life. But the question of how chromosomes came to exist and evolve has long been discouragingly hard to answer. This is due partly to the lack of chromosome-level genomic information, and partially to the suspicion that long periods of evolutionary change have washed away clues about that ancient history. And then in this paper, in Scientific Advances, this team, they've been able to track changes in chromosomes that occur as much as 800 million years ago. They identify 29 big blocks of genes, so that those blocks that I showed you, like A, B, C, D, that's a block. And so those blocks, they were able to identify blocks like that. And they remain recognizable as they passed into the 3 earliest subdivisions of multicellular animal life. So this is, your different parts of the tree, like, you have plants and animals for multicellular organisms, as well as your, so you have basically plants and animals. Using these blocks as markers, the scientists deduced how the chromosomes fused and recombined as those early groups of animals became distinct. The researchers call this approach genome tectonics, in the same way that geologists use their understanding of plate tectonics to make sense of the movement of continents. These biologists are reconstructing how various genome duplications, fusions, and translocations create the chromosomes we see today. So this is something that they're thinking is a new tool for comparative genomics. So if you want to compare genomes from one organism to another, this is a good way to look at what's conserved and what's not. And this is not just for closely related organisms, but the difference between insects and worms and humans. So you can actually track a lot of these. And this is important because we're starting to get chromosome level genome assemblies. So once we get these kinds of data, we can actually look and verify and see what the gene order is in a lot of these organisms. So we can use that information to make statistical predictions and rigorously test hypotheses about how groups of organisms are related. So this is a tool to understand evolution a bit better, but also development, because these blocks of genes that we have here contribute to developmental order. So, the different segments of the organism, but also kind of, these processes are governed by genes that are linked together. So A and B might be linked functionally. They might, B might be dependent on A for something to produce, say, like an enzyme or some sort of product in the cell. If B and A are broken apart, then that can no longer happen. So that function is gone. And that becomes something that can be deleterious to the organism. It can kill the organism in development. So this is an important thing, this physical linkage. But physical linkage over a little bit longer scales, usually if 2 genes are next to one another, they're linked in the sense that they don't get pulled apart often, because recombination is something that happens probabilistically, but also as you get away from if you get away from A, D is more likely to be recombined away from A than B. So those blocks are somewhat stable over different distances. We don't really understand if whole big blocks can be conserved or kept together by any mechanism, but we know that recombination is something that happens a lot in genomes. So that's a thing that can happen. So they're able to, this is an important part of genomes and of evolution and of development. So using similar innovative methods, this group resolved a long-standing mystery about the timing of genome duplications that accompanied the emergence of jawed vertebrates. So these are we have jaws, and there's gnathostomes is the group of organisms that have jaws. So this includes, your mammals, some birds, and I think it goes into fishes a little bit, but anyways, there's this group of organisms called gnathostomes, and these are the jawed vertebrates. But the importance of the approach isn't purely retrospective. In the process of making these discoveries, the researchers are learning about the algebraically simple rules that govern what happens when chromosomes swap chunks of themselves. That information can guide future genomic studies and help biologists predict when they'll find in the genomes of the species not yet sequenced. So they're actually looking for rules in this data. So again, we don't know the rules that govern Synteny. We don't know the rules that govern some of the, how these things are kept together or broken apart. We only know that they're generally probabilistic things that it happened, but we don't really know much more about it. we're starting to get a broader picture of chromosome evolution across the tree of life. And now we can actually understand this a little bit better because we understand these kind of processes. And then we can attach rules to them, perhaps, if we have the data, if we can build computational models of them. So what do ancient genomes share? They actually looked at the genome of the Hydra, which they describe as a model of a venerable cnidarian. By comparing it to the available animal genomes, they discovered highly conserved groups of linked genes. So Hydra is a marine invertebrate that's they're using as sort of like the baseline for this, or the common ancestor. And they're looking at all these animal genomes, and they're looking across those genomes to see what's shared. And the idea being that whatever's shared is conserved amongst all of them, or something that doesn't change amongst all of them from the common ancestor. Although the order of the genes within a block are often shuffled. So that means that when you have these genes are shuffled like this, but they don't break apart and go to other blocks or other parts of the genome. So that means that they're shuffled, but they're not broken apart. So there might be some way that they maintain their integrity, functional integrity, but it's a different way. And you might expect that if you have different organisms, like if you have the difference between a hydra and like a cow, there are, of course, differences there that, maybe the genome is mediating. And if you shuffle these genes within these blocks, then it becomes, you can see that that's a source of diversity. The blocks themselves are stable over long stretches of time. And so this is something that, we talk about like mutations in genes. We talk about proteins. We talk about that. We never really talk too much about the larger scale things in the genome, like these Synteny blocks or chromosomes themselves. But there's a lot to understand about that, and that's a lot of the diversity between species as well. So this is an interesting paper. Then there's this idea of genomic inertia. But what would cause the blocks of genes to stay linked together? There's a Synteny idea that I just explained. So that's the mechanism that they're proposing here. So, and then this paper here is actually one of the papers from this area of research. And so Synteny-based analysis indicates that sequence divergence is not the main source of orphan genes. So this is where you have the sequence divergence, and they're using Synteny-based analysis to look at it. So they're looking at orphan genes, which are species-specific sequences that lack detectable relations in other species, and then they're saying that they can use the Synteny-based analysis to show that sequence divergence isn't the source of orphan genes, or that they didn't evolve out of like multiple mutations. They evolved out of something else. And so this is an example of how they use Synteny to do, to look at evolutionary relationships. So that's all I'm going to talk about today. Thanks for attending, everyone. Jiahang and Harikrishna, ABD, thank you for attending. Does anyone have any questions at this point about anything before we go?
Hi.
Hi.
No questions from here.
Okay. So we'll this will be on YouTube, so you can see the whole thing if you have questions about GSoC or other things. And so part of the GSoC experience over the summer is, during the program, is to attend the meetings, and you'll be able to demo your project weekly. So we'll have a period of time at the beginning where you can show your project and you can show your progress, and then we'll have questions about that as well. And then you'll be able to contribute in other ways as well. So if you're working on the project, though, I would recommend that be your first priority. But anyways, just wanted to wish everyone a good week. If you don't have any other questions.
left for this project? Just one.
Probably at least one. It really depends on how many slots INCF gets. So in the past we've had one, but we could have two. It really depends. I hope that we get one. We applied as an organization and we didn't get in, but that was just to maybe facilitate more positions. But anyways, it usually is somewhat competitive. So but I think the best strategy is just to have me review your proposal or have Ujjwal review it, and that'll help.
Thank you.
Okay,
And thank you for the meeting.
you're welcome. Thank you. Okay, have a good week.